Hello guys, welcome to another video. Today we will see the main characteristics of Monera Kingdom. In here, I need you to really remember the history of taxonomy. First, remember that Robert Whittaker divided these 5 biological kingdoms and established the name of Monera to talk about bacteria. But later on, Carl Woese divided the kingdoms, in this case Monera Kingdom, into 2 main groups, which are Archaeas and Eobacteria. This is where we're going to focus our attention on in this video, the difference between Eubacteria and Archaea, or as Robert Whittaker firstly described them, Monera Kingdom. Use this list to make your Cornell's method notes. Are you ready? Let's go. First, let's start by making clear the difference between Archaea and bacteria. Archaeas are known as extreme organisms. Why is that? Well, firstly, because scientists found them on these geysers, these hot spring lakes on Yellowstone first, and they firstly thought that they were bacteria, but no real bacteria could be found on these very hot springs. A closer analysis of these organisms revealed that they were more related to eukaryotic organisms than actual bacteria. So therefore, a new group of these ancient or extreme bacteria was made, the Archaeans. Actually, Archaeas are a possibly link to found living creatures in another planets because they can endure very harsh conditions. On planet Earth, Archaeas could be found in very salty places and also in places without oxygen, in places where no other organism could. On the other hand, bacteria could live almost everywhere, on the soil, on the water, and also inside and out organisms. They could be part of the normal flora of microbes that lives inside and outside you, or also they could be parasitic bacteria producing diseases in many organisms. Well, focusing on our first characteristic, bacteria and archaea are all unicellular organisms, which means that they are formed by just one cell. The shape could be really different. They could be called cocci, bacilli, or they could have very weird screw shapes, like you can see here on this slide, right? But remember that they are all made of just one cell. In this case, we don't have multicellular organisms. Focusing a little bit more on the cellular structure of these bacteria and archaea, they are considered prokaryotic cells, which means that they don't have nucleus nor organelles. Now, where is the genetic material found if there is no nucleus inside? Well, the genetic material is just spread inside the cytoplasm. And one main thing of the genetic material is that it is a circular shape. Instead of being a linear molecule of DNA, like the one, for example, that we have inside our eukaryotic cells, DNA in this case is a circular molecule. Another important thing is that besides that circular molecule of DNA, bacteria and archaea could have plasmids, which are small fragments of DNA in a circular way that are dispersed on the cytoplasm as well, and they are very important for the reproduction of bacteria. Another important structure that covers the cell, in the case of bacteria and archaea, will be the cell wall and the capsule. Now, archaeas and bacteria are a little bit different depending on what make these layers. In the lower section or just near the cytoplasm, we will have the cell membrane. Then we will have the cell wall, which in archaea and bacteria, the composition will slightly differ. In the case of bacteria, this cell wall, the yellow layer that you're seeing on the diagram, is made of a substance called peptidoglycan, while in archaea, that cell wall will be made of another substance, not peptidoglycan. An extra third layer is called the capsule. Some bacteria may have it, some bacteria may not, but in this case, you should remember that they could have these 3 layers. If we want to be really specific about where this cell wall is found and how is it made of, well, we can see that the bottom section, the most internal layer is the cell membrane, made usually as the eukaryotic cells membrane is made of, phospholipids, cholesterol, and protein. And above it, we will have a thick layer of peptidoglycan only found in bacteria. Archaea, remember, they don't have peptidoglycan. Another important characteristic of prokaryotic cells, or in this case bacteria and archaea, is that they don't have organelles. Remember that organelles are all the membrane-bounded inner structures with a specific function. The only kind of organelle that bacteria and archaea have are the ribosomes. Remember that ribosomes are very important for protein synthesis, are those tiny factories of proteins in which RNA should be translated into proteins. In this diagram, ribosomes should be seen as these tiny little blue dots. That's the only organelle that bacteria and archaea will have. Now, in the external side of bacteria, we can find the flagellum. Flagellum are, as in any other cell, needed to move the cell. Now, some projections that could be sticking out of the capsule and also as part of the membrane are called the pili. Pili are needed to attach the bacterium to the surface in which bacteria could live. Now, pili could also have a second function, which could be used into kind of a sexual reproduction form that we will call conjugation. We will talk about it a little bit later. Talking about the type of reproduction, bacteria usually reproduce asexually, which means making clones by a process called binary fission. Binary fission is very similar to mitosis. So that's the way the cell divides into two copies and therefore could colonize any place. Now, as I said, some bacteria could have a second type of reproduction, which is similar to sexual reproduction, a kind of sexual reproduction version called conjugation. To do that, the bacteria uses a special type of pili called sex pili. That sex pili is usually used to attach or to catch another bacteria and then create like a kind of membrane bridge. And using that bridge, the bacteria could share pieces of plasmids. Remember that those plasmids are small DNA molecules that could easily be shared between bacteria using these membrane bridges. This is how bacteria usually share genes, which is kind of a sexual reproduction version. Now, once bacteria had reproduced, if the conditions are not ideal, they could form a bacterial spore or also called an endospore. This endospore, it's a very resilient structure that could remain dormant, which is kind of sleep until the conditions are ideal again and the organism could regrow. Some scientists even believe that these endospores could be found on the surface of other planets, and that could possibly expand the way in which we could study life outside our planet. Now, let's talk about the metabolism of these organisms. Remember that metabolism are all the chemical reactions that organisms do to produce and use energy. In this case, respiration and nutrition. Talking about respiration, Monera Kingdom or Archaea and Eubacteria could use oxygen. Therefore, they could be considered aerobic organisms. Remember that oxygen is needed to produce ATP. Or they could be also organisms that don't require oxygen, also known as anaerobic organisms. The way in which these anaerobic organisms will produce its ATP is by a process called glycolysis. Now, talking about nutrition, bacteria and eubacteria, also archaebacteria, could be known as autotrophic organisms. Some of them could produce their own food by means of photosynthesis, meaning using sunlight. Others could produce their own food by requiring chemical substances, therefore being known as chemosynthetic organisms. On the other hand, there could be bacteria, archaea, and eubacteria organisms that feed on other particles, or feed on substance produced by others, therefore considered heterotroph organisms. Now, in this section, they could be found bacteria of free life, which they will be found on the soil, on the water, also having the function of decomposing organic matter, or they could be also parasitic bacteria, which are the bacteria that commonly produce disease on the organism. Why is it important to study bacteria and archaea? Well, especially bacteria are important because they are the main recyclers of dead matter. Um, some people may say that if we didn't have bacteria and also a group that we will see a little bit further, which are fungi, we will be filled of dead matter. They're bacteria and fungi, the main recyclers of organic matter. They return the nutrients to whatever they found it. Now, bacteria could also be important because they produce many diseases. Remember that not all bacteria are parasitic, but well, in the case of humans, we can see many human diseases produced by bacteria. Now, another important thing is that bacteria could be used as biotechnological organisms or biotechnological tools. For example, recombinant insulin is produced by bacteria. Also, bacteria could be used in fermentation to produce yogurt or cheese. And also, bacteria could be used to make genetic studies. So, this is a very short kind of summary that I wanted to share with you so you can remember the structure, the importance, and the function of bacteria, archaea, and in a general speaking manner, Monera Kingdom. Remember, love biology and remember to study a little bit further this topic if you're interested in it. See you later, guys. Goodbye.